ड्रग डिस्कवरी का है तो अच्छा।
Hello.
Hello, sir. Good afternoon,
Madam,
sir.
is it audible?
Yes, sir. You're audible and visible.
Okay.
Sir, shall we start the program?
Hello, good afternoon, Amma.
Good afternoon, sir. Sudarshan Reddy, sir. Shall we start the program, sir?
Yeah, you go ahead.
We have Umesh sir and Lalita madam also.
Okay, fine.
Distinguished Chief Guest, Professor U. Umesh Kumar, sir, Head Department of Chemistry, Usmania University. Guest of Honor, Professor Lalita Guru Prasad, Madam, School of Chemistry, University of Hyderabad. Professor K. Mutyam Reddy, sir, Secretary HMVS. Professor Sudarshan Reddy sir, Secretary and Correspondent College, below Principal Dr. J. Achita Devi Madam, Vice Principal Dr. C. Kavita, coordinators, HODs, colleagues, and dear participants, a very good afternoon. I, Dr. Jeevan Jyoti, Head Department of Chemistry, feel honored and privileged to welcome you all to the inaugural session of 1 week national level faculty development program on role of ADMET and molecular docking in drug discovery, organized by Department of Chemistry in collaboration with Q Statics Private Limited, Hyderabad. The discovery and development of new drugs is a lengthy and complicated process, which demands significant time, energy, and resources. The drug candidates have a high attrition rate as they often fail to show the anticipated efficacy, and sometimes they are found to pose unacceptable risks to human health. ADME tox studies play a vital role in evaluating the pharmacological properties of a drug candidate that can finally make it a success. Acknowledging the importance of ADME and tox studies, drug discoverers have already started to undertake them at the very early stages of the whole process. The main aim of conducting this 1 week national FDP is to provide a basic platform to the faculty and research scholars to attain an immense knowledge about prediction and evaluation of ADMET and molecular docking in the field of drug discovery. I hope all the participants will have the 6 most productive days of interesting and informative deliberations by the invited speakers and hands-on by the expertise of Statics Private Limited. I sincerely wish that the FDP will be successful in reaching its goal as mentioned and assure you of the full support from all of us. And I wish you all the participants a happy learning. Now, I invite Ms. Shravanti, Assistant Professor in Department of Chemistry, to proceed further.
Thank you, ma'am. Good afternoon to one and all. I, D. Shravanti, Assistant Professor in Department of Chemistry, RBVR Women's College, welcomes you all to this 1 week online National Faculty Development Program on role of ADMET properties and molecular docking in drug discovery, organized by Department of Chemistry, RBVR Women's College, in collaboration with Q Statics Private Limited. Now, the session starts with a prayer song by Satvika. Over to you, Satvika.
गणपतये नमो नमः। गणपतये नमो नमः। गजवदना गणनाथ प्रभो, गजवदना गणनाथ प्रभो, संकटहरणा भवभयहरणा, संकटहरणा भवभयहरणा, वेदविनायक मोदकस्था, विद्यादायक हे मम बन्धु, गजवदना गणनाथ प्रभो प्रभो। चिन्मय रूपा विश्वाधार, चिन्मय रूपा विश्वाधार, पंकजलोचन हे गणनाथा, शंभुकुमार गौरी तनया, शंभुकुमार गौरी तनया, गुणगण निलया पालय मां, गुणगण निलया पालय मां, गजवदना गणनाथ प्रभु प्रभु।
Thank you, Satvika. As our principal madam, Dr. J. Achita Devi, is suffering with fever, severe cold and throat infection, ma'am is unable to speak. Hence, we request our vice principal madam, Dr. Kavita madam, to address the participants.
Am I audible, Shravanti?
Yes, ma'am, you are audible.
Yeah. Professor Sudarshan Reddy sir, Secretary cum Correspondent RBVR Women's College, our Chief Guest Umesh sir, Guest of Honor Lalita ma'am, members from Q Statics, team of Chemistry and Forensic Department, the participants, and dear colleagues of RBVR Women's College. I extend a very warm welcome to all of you to this inaugural session of the 1-week online FDP that is organized by the Department of Chemistry and Forensic Science. It gives me immense happiness that the Department of Chemistry is at the forefront in organizing such activities, which would not just enrich the teaching fraternity, but would also build a system or a culture of collaboration between the industry and the academia. So let me talk about RBVR Women's College. Let me tell you all briefly about the institution that I represent. So RBVR Women's College was established in the year 1954, Hyderabad Mahila Vidya Sangam, which is a non-profit educational society whose primary vision was to empower women through providing education to them. Now, our college happens to be the second oldest women's college in the city of Hyderabad. The college owes its existence to the foresight of Sri Raj Bahadur Venkat Rama Reddy, who was the Kotwal of the erstwhile Hyderabad state. He strongly believed that education is the only way that women can be empowered. His vision was also supported by many other luminaries, like Sri Burugula Ramakrishna Rao, who's the first chief minister of the erstwhile Hyderabad state, Sri Madapati Hanmanta Rao, who's former mayor of Hyderabad and chairman of the Legislative Council of AP, and Sri K.V. Ranga Reddy Garu, former deputy chief minister of AP. He was supported in his efforts by all these luminaries. Even till date, this legacy of noble service is continued by the members of the governing council of our college, who comprises of judges, professors, and other people of eminence, all rendering their honorary services. Our college had the rare privilege of welcoming Pandit Jawaharlal Nehru, the first Prime Minister of India, who laid the foundation stone for our college building. Dr. S. Radhakrishnan, the former President of India, inaugurated the college building, and Srimati Indira Gandhi, former Prime Minister, inaugurated the hostel building. When the college celebrated its Golden Jubilee celebrations, Dr. APJ Abdul Kalam, the former President of India, addressed the students, which is again another landmark in the history of our college. The valedictory program of our college, Diamond Jubilee celebrations, was graced by Sri C.H. Vidyasagar Rao Garu, who was the then governor of Maharashtra, and Her Excellency Dr. Tamilisai Soundarajan, governor of Telangana, was the chief guest for the convocation in 2019. Our college is affiliated to Osmania University, and it is also recognized by UGC. We were conferred autonomous status by UGC in the year 1989. We have been accredited by A grade by National Assessment and Accreditation Council from 2007 to 2017. And in 2018, that is in the fourth cycle of re-accreditation, we were awarded B++. Our college is one of the best infrastructure in the city of Hyderabad. Our college is centrally located. We have a 3.5-acre campus. We have 6 buildings, 50 classrooms, 30 science laboratories, staff rooms, and also an in-house hostel. Presently, we're offering 17 UG programs and 5 PG programs. Apart from these, there are several life skill courses, career-oriented courses, skill enhancement courses, and certificate courses that we are also offering. Our college has fully functional NCC and NSS units. Our faculty strength is more than 100. We have close to 30 teachers with PhD degrees. We have more than 20 teachers who have NET set certification. We have 3 faculty members who have MPhil degrees. We have a number of our faculty members who are working on research projects funded by various bodies. So these are all the academic and infrastructural benefits that we offer to our students. Thank you.
Thank you, ma'am. Thank you so much. Now I request our Honorary Secretary and Correspondent, Sir Professor G. Sudarshan Reddy, sir, to say a few words on this occasion. Over to you, sir.
Thank you, Sravanti. I'm very happy to be part of this inaugural program of 1 week National Faculty Development Program organized by the Chemistry and Forensic Science Department of the college in collaboration with Statics Private Limited, Hyderabad. The members from this Q Statistics, Dr. Purushottam and Dr. Karunakar. I welcome them on behalf of the college. And at the outset, I welcome the two distinguished guests, Professor Umesh Kumar, Head Department of Chemistry, Usmania University, who is fresh from organizing a 2-day international seminar, and I congratulate him for that. And I also welcome Professor Lalita Guru Prasad from the School of Chemistry, University of Hyderabad, as the guest of honor. And looking at the program of the week, I find it very interesting, and looking at the resource persons and themes drawn from the Nagarjuna University, the Naipar Kolkata, the PGI Chandigarh, then the other 2 sessions being conducted by the Purushottam, Dr. Purushottam and Dr. Karunakar. And I congratulate Dr. Jeevan Jyoti, Head Department of Chemistry, and her teammates for organizing such programs. And I think this program will help the faculty and the senior research scholars to enrich themselves in the subject of chemistry and in various nuances of the chemistry subject. And I once again thank the Department of Chemistry for inviting me to be part of this inaugural session. And I once again welcome all the distinguished guests and the participants to this program. Thank you one and all, and thank you very much.
Thank you, sir. Now I request HOD ma'am, Dr. V. Jeevan Jyoti, to speak about the department.
Thank you, Shravanti. Our Department of Chemistry was started in 1954 with 59 intermediate students. It has now grown to be the largest department with 800 undergraduate, 60 postgraduate students, and 19 well-experienced faculty members, which includes 2 research supervisors. It is also the nodal department for forensic science subject. Besides BSc, MSc, and PhD programs in chemistry, we offer 9 certificate courses. The students are trained in well-equipped teaching laboratories and a centralized instrumentation research laboratory, which is well-equipped with UV visible FTIR and fluorescent spectrophotometers. We are also happy to inform that we are also providing analytical services to other colleges. The department has 16 MOUs with research and academic institutions. Our department is recognized research center by Usmania University since 2017 for conducting research leading to the award of PhD degree and also for MSc student project dissertations as part of the curriculum. We regularly conduct seminars, training programs, workshops in order to upgrade and enhance the standards of teaching and learning. We also have a student faculty club, Curie, in order to promote experiential and participative knowledge to our students. With this short introduction, I once again welcome all the members to the faculty development.
Thank you, ma'am. Now I request Dr. B. Anupama Madam, Associate Professor, Department of Chemistry and IQAC Coordinator, to introduce today's chief guest, Professor Umesh Kumar Sir, Head Department of Chemistry, University College of Science, Usmania University. Over to you, ma'am.
Thank you, Shravanti. Good afternoon, everyone. It gives me an immense pleasure to introduce today's chief guest, Professor U. Umesh Kumar, Head Department of Chemistry, University College of Science, Usmania University, and Dean, Faculty of Sciences, Palamuru University. Professor Umesh Kumar Sir completed MSc and PhD from Usmania University Sir has done postdoctoral degree from University of Vermont, USA. Sir has 32 years of teaching and 25 years of research experience. He has published 30 research papers in various national and international journals. Sir presented papers in international conferences. His research areas are blood coagulation factors, protein analysis, different instrumentation methods for protein analysis, etc. Sir's awards and achievements are, Sir got first rank in MSc Chemistry and awarded by Senior Research Fellowship by CSIR Government of India. Sir is a member of American Chemical Society, and Sir received Indira Gandhi National Level NSS Trophy from the President of India. Sir held many positions in Osmania University, Mahatma Gandhi University, and Shatavahana University. Few of them are Joint Director, Directorate of Admissions, Osmania University, Chairman, Board of Studies, Department of Chemistry, Osmania University, Registrar, Mahatma Gandhi University, Nalgonda, Registrar, Shatavahana University, Karimnagar. Recently, Department of Chemistry, Osmania University, organized international conference successfully under Sir's dynamic leadership. With this brief introduction, I would like to invite Sir to address the gathering. Thank you one and all. Sir, over to Umesh Kumar, sir.
Is it my voice is audible, madam? Hello, my
Yes,
voice
sir.
is
Yes,
audible.
sir.
Yes,
I'm audible,
Yes, sir.
madam.
yes,
Yes,
sir.
sir, it is
audible?
audible.
Yes, sir.
So can I start?
Yes, sir, you can start.
So good afternoon, everybody. So I would like to welcome all of you for this 1 week online nationaries marker docking. And first of all, I would like to congratulate the Department of Chemistry for organizing this 1 week online program. And I know very well Professor Sudarshan Reddy Garu, correspondent of Reddy College, and Muttam Reddy Garu, and Principal Madam, and Jeevan is the Head Department of Chemistry, and research scholars who are present in this online program, and MSc students and other faculty members of Reddy College and other from other colleges. So first of all, I would like to congratulate the Department of Chemistry for organizing this wonderful 1 week program, and let me thank organizers for inviting me as chief guest of this function. Okay, thank you very much for one and all for inviting me. And very recently, just day before yesterday, that is on 29th and 30th, we organized an international seminar in the department conference, and we got a very good response for all the members. Actually, I advised the Madam John Jyoti Madam and Principal Madam to organize this conference offline, but I don't know what is the reason, because now the pandemic situation is not that much around the global and as well as in our state. So we have organized the program offline. But next time, anyhow, this happened is happened. So my advice is next time better to conduct this program offline. And at the same time, today's guest of honor, Lalita Guru Prasad, Professor Lalita Guru Prasad from School of Chemistry, University of Hyderabad. So I would like to welcome Professor Lalita Guru Prasad also. Okay. So the thing is, any international seminar or national seminar, it will aims to provide an opportunity for desired interaction among the students, researchers, and academicians working different areas on chemical sciences. The main objects of any seminar will be to enhance the knowledge, promote scientific temper, and to highlight the recent trends and issues in the field of research in chemical sciences. I hope this one seminar, all these things, and it is expected that this seminar will contribute to update knowledge in the field of these branches and will encourage the young researchers for promoting newer research endeavors in the challenging domain of modern science. I think this seminar also provides a platform to investigate problems in the interdisciplinary fields leading to collaborate research. The seminar will embrace keynote address, lectures, plenary lectures, and invited lectures. And I personally, on my behalf and on behalf of the Department of Chemistry, I extend a warm welcome to all the keynote speakers, conference speakers, research students, master students, invitees, and everybody. So thank you to each and every one of you for being present here today. And the COVID-19 pandemic has sent shockwaves through the globe, and the dust is settling down slowly. So the last 2 years have been very difficult for many of us here. And we thought this is the right time to continue our scientific endeavors through this conference. According to Richard, a Nobel laureate, chemistry in 1996, the global top 10 problems for next 50 years are energy, water, food, environment, poverty, terrorism, diseases, education, democracy, and population. This conference is likely to address energy, water, and environmental problems and other problems, and particularly in the field of molecular docking in drug discovery. So this conference, the main aims of this conference to bring top-class academicians, young researchers, young faculty and students under one umbrella to exchange scientific ideas. And this also provide a platform to young research students to present their work in the form of, I think if there are any poster or bold presentations, they can present their work. And it also provide an opportunity to research students and faculty to listen lectures by eminent scientists on cutting edge research topics. I hope that this 1 week will be full of deliberations on different fields. And at the same time, I'm confident that our young research and master students and faculty will be benefited by these deliberations. So I hope this 1 week program may be a grand success. And once again, I thank all the organizers for inviting me as chief guest. And at the same time, I would like to congratulate all the members, all the organizers, and particularly the correspondent, secretary, Reddy College management members, and faculty members, head of the Department of Chemistry, for organizing this program. So all the best to everybody. Thank you one and all.
Thank you, sir, for your valuable message. And also we take your suggestions, sir. Next time onwards, we will arrange offline program in future. Thank you so much, sir.
Okay.
Now I request Dr. P. Jansi Lakshmi Madam, Assistant Professor, Department of Chemistry, to introduce today's guest of honor and keynote speaker, Professor Lalita Guru Prasad Madam, School of Chemistry, University of Hyderabad. Over to you, ma'am.
Thank you, Shravanti. Am I audible?
Yes, ma'am, you are audible.
Yeah. I feel honored to introduce today's guest of honor and keynote speaker, Professor Lalita Guru Prasad ma'am from Hyderabad Central University. Ma'am holds MSc and PhD in chemistry from Osmania University, Hyderabad. She has carried out post
Ma'am, you are not audible.
Now it's okay?
Yeah, ma'am, it is audible
Yeah.
now.
Yeah, repeat. Madam had carried out postdoctoral work in Birkbeck College, University of London, Department of Biochemistry, University of Cambridge, UK, and CCMB, Hyderabad, India. Madam joined the School of Chemistry, University of Hyderabad as lecturer in October 2000 and is a professor since 2010. Madam's group is pursuing research to understand protein sequence to structure and function correlation. This is achieved by computational studies, protein sequence analysis to identify novel repeats and domains. Madam has also expertise in protein three-dimensional structure modeling based complete proteome function analysis, molecular docking, quantitative structure-activity relationship studies, and molecular dynamic simulations to explain the molecular basis for function. Madam's research emphasizes is on protein in M. tuberculosis, H. pylori, and human kinases. Some of the hypotheses from computational studies for M. tuberculosis proteins are validated experimentally. Purification and characterization of low molecular weight proteins from plant sources are also being studied in her lab. Now, I invite ma'am to deliver her keynote address on the topic relevance of ADMET properties and molecular docking in computer-aided drug design. Ma'am, over to you, ma'am.
Thank you so much. Can I share my slides? I'm audible,
Yes,
right?
ma'am. Yes, ma'am. Yes.
Host disabled
Hello, ma'am.
participant screen sharing.
Hello,
I think you should permit me to share my screen.
madam. Hello,
Can
madam.
Can you hear me?
Hello, madam.
Can
Hello.
you hear me?
Yes, ma'am, you're audible, but first you give your message, ma'am. Later we can present your slides.
Okay, fine. I thought I could club them together. I would say that I'm very much honored to be a part of this, elite group of faculty, students, research scholars. I'm also Osmanian. I've done my MSc and PhD as Jhansi ma'am earlier said. What else? I would say that, COVID has taught us many things. COVID has taught us not only to, to keep ourselves safe, but then COVID has also taught us how to be able to do our work very diligently in spite of, the restrictions that we had. So I hope, this will be one of the last lectures, at any place where, things are being conducted online. And as I said, I thank you very much for having me, on board. And I congratulate the RVBRR College for conducting this, week-long faculty development program. Actually, I personally feel that the faculty development programs are better in one sense that, like, if you train a teacher, it's like training several batches of students because they're able to disseminate the knowledge to many more, batches or generations of students to come. So I think, this is one way, whereby the faculty not only improve their, not improve, they just brush their knowledge. They already have this knowledge. It's only a matter of brushing their memory once again to come on board, and they're able to disseminate the same knowledge to the, to the future generations. And, like I was telling you about COVID earlier. So during COVID, what we have seen is that, there was a great need to discover vaccines. That's one part. And in addition to, in addition to, coming up with new vaccines, the inventions, there was also a great need to come up with new molecules as drugs. But the drug design pipeline per se takes a long time. And if you read any, research article, it says that drug design pipeline takes at least about 10 to 20 years from finish to end. But what people could do during COVID times was, come up with old molecules like favipiravir, remdesivir, so on and so forth, and say that these molecules are also, reasonably good in order to combat, COVID-19. In a situation when we did not have anything, when people could come, researchers could come up with some suggestions. This has happened because of the intervention of experiments. Of course, experiment is the ultimate truth. But initially, what the researchers have done is to take the existing molecules and see how well they fit into the protein targets of the SARS coronavirus 2 proteins. Okay, so that's one way we can say that, we have been making impact of computer-aided drug design in the last 3 decades. But the last 2.5 to 3 years, we have seen an overwhelming response of researchers using these tools to come up with, either new molecules or to come up with, the already known molecules for newer diseases. And again, as I said, experiment is the truth, but these tools, computational tools, definitely will add a value in giving a direction to the project, in giving a direction to the way the project has to be taken, which molecules have to be dropped, which molecules could be picked up, because I mean, basically it brings down the cost. It also brings down the time taken for a new molecule discovery. Okay, so with this, I would say that this is definitely a tool that is there to stay with us. And of course, as the theme of your faculty development program is, the classical CAD might be taking a backseat, but now with AI and ML, there are more tools available for for coming up with better models. And all the participants who are sitting here should know that there is a method called as AlphaFold, and this method is able to give these structures of proteins. Like protein structure modeling was an art, it was a science, but now it has become a child's play. Like you're able to model the structure of a protein effortlessly, and these structures are available for 200 million proteins at EBI from a database. So all the students, I mean, the participants of this program, I recommend them to go back to EBI and make use of these models that are already available for the protein structure. So with this, ma'am, I thank you once again for giving me this opportunity of being in this August gathering.
Thank you, madam, for your wonderful and valuable message. From Department of Chemistry and Forensic Science, we would like to present a letter of appreciation to our Honorable Chief Guest, Professor U. Umesh Kumar Sir, Head Department of Chemistry, University College of Science, Usmania University. Please accept, sir, as a token of gratitude. Hello.
Okay, madam. Thank you very much. Thank you very much, madam.
Thank you, sir. Now I request Dr. Umaima Ghazal, Assistant Professor in Department of Chemistry, to propose formal vote of thanks for inaugural session. Over to you, ma'am.
Good afternoon, everyone. This is Dr. Umaima Ghazal from Department of Chemistry and Forensic Science, feeling delighted to offer vote of thanks on 1 week online DP on role of ADMET properties and molecular docking in drug discovery. I'm extending my sincere thanks to our chief guest, Professor Umesh Kumar, head of the Department Chemistry, Usmania University, our guest of honor and keynote speaker, Professor Lalita Guru Prasad, School of Chemistry, University of Hyderabad, and other reverend speakers, research scholars, faculty members, and participants. I would like to encompass my sincere thanks to our honorable chief guest, Professor Umesh Kumar, who emphasized the interaction of chemical society members and to enhance their knowledge in various fields to protect environment in the best possible way. Professor Guru Prasad also signified the value of FDP among researchers and academicians so that they can develop some path to fight against COVID-19 as well. I want to acknowledge all the esteemed members on the behalf of the entire Department of Chemistry, of management of our college, including respected Professor Nitin Reddy, Professor Sudarshan Reddy sir, our Venerable Principal Madam, Dr. Achita Devi, Vice Principal Madam, Dr. Kavita, IQAC Coordinator, Dr. B. Anupma, and to infuse us to conduct such edifying sessions. With deep sense of appreciation, a great thanks goes to our HOD, Dr. B. Jeevna Jyoti, for her determined efforts. I also want to thank all the people who became the part of this 1 week online FTP. Thank you all for sparing their pleasure time with us. I'm concluding with this hope and remark that all participants absolutely on opposite information on ADMET properties and molecular docking in drug.
Thank you. Thank you, ma'am. Dear participants, in 2 minutes, we will start the first session by our guest of honor and keynote speaker, Professor Lalita Guru Prasad, Madam, School of University of Hyderabad. Now, I request Madam to give her presentation. Over to you, ma'am.
Yeah, thank you, ma'am. I'm just trying to share my screen. I think you should permit me to share my screen.
Okay, ma'am. Yes, ma'am, you can present now.
Let me just try that. Sure. You cannot see my presentation still, right?
Yes, ma'am, it is not visible.
One second, yeah, I'm just going over there. Now it should be coming.
Yes, ma'am. Ma'am, you keep slideshow, ma'am.
One second. I'm a little confused. No, correct. Now it's coming.
Yes, ma'am, it is visible.
I need to do something else, I should say. Hide or something. Your screen is being shared. Okay, fine. Yeah. Yeah. As I said, once again, I thank for having me on board to share my little bit of experience in working on molecular docking and ADMET properties, of course, are a part or parcel of the new ligand discovery. The title of my presentation is Relevance of ADMET Properties and Molecular Docking in the Computer-Aided Drug Design. There is something that is masking my top part of the screen. Okay, right. So what I would be talking to you about is basically, I'll actually open my presentation in the other computer because I'm only able to see half of my screen.
But ma'am, it is visible, ma'am. For us,
It's visible to you, but for me,
yes,
there is a right panel with myself and some more names. On the top, it is mute, stop video. Those things are taking a lot of my time. And
ma'am, you can minimize that.
Yeah, I could minimize them.
Yes, ma'am.
Yeah, that has worked, yes. Right. So yeah, I would say that we are here to learn a little bit about some of the modules or pipelines in the drug discovery, so one of the I mean, if you go from first to last of the pipeline in the drug discovery, the first would be to identify the protein target or a receptor target to which you would want to identify a small molecule. Once you identify that, once you identify what is the receptor target for which you want to design small molecules, the second step would be to find out what are your basket of small molecules about which you will work with in order to be able to see if they're able to bind to the receptor target. And having identified them, then you will do the matching between the receptor and the small molecules. That is, one will go ahead with the molecular docking to find out which molecules fit to the receptor in the active site of the receptor or in the overall allosteric pockets of the receptor. And having established that, you would want to find out whether this molecule is able to bring about any required therapeutic effect. That of course requires experimental methods. But before you go to the experimental methods, because your molecular docking will report several hundreds and thousands of molecules, and because it is also difficult to work with hundreds and thousands of molecules, it is good to intervene and use certain computational methods to find out what are the pharmacokinetic properties of the molecule, such that the molecule will actually end up being hopefully a good drug molecule. Okay. So in the drug discovery pipeline, molecular docking and the pharmacokinetics parameters such as the ADMET properties is what I will be discussing with you. So in a classical drug discovery pipeline or a project, the main aim would be to identify a new molecule, and this new molecule should be hopefully becoming a drug molecule in future, such that we are able to take this molecule as a medicine sometime in the near future. And as I said in my introduction, for over 3 decades, the computer-aided drug design tools are being used in the drug discovery pipeline. And in the last 2.5 to 3 years, this has become much more robust, and there is an increase of several fold in terms of number of papers being published using CAD methods during the COVID times. And this method is here to stay, and this has in fact become a indispensable tool by chemists and biologists all over. And also this CAD methods will tell you if you already have a lead molecule, or if you have a good molecule, then this CAD methods will actually help you also to refine a molecule. So it's not just the new drug discovery, but then if there is an existing drug, then how to beat the patent such that you're able to come up with new molecules can also be done using the CAD methods. And yeah, so as I said, one of the things that I'll be talking to you about is about the ADME properties. And ADME properties will allow the drug developers. ADME properties will actually allow the drug developers to understand the safety and efficacy of the molecules. All right. So, what do you mean by safety and efficacy of the molecules? If this drug is taken by an individual, even before it becomes a drug, if it's at the development stage, if this a molecule which is at the development stage, if it's given to an individual, is it safe? Is it efficacious? Will it show the desired effect? Are the questions that are being answered by the ADME properties. And molecular docking will tell us whether this particular molecule in question is able to bind to the target receptor, and therefore okay, even when it binds, does it bind in a weak manner, does it bind in a strong manner? That is the binding affinity between the protein and the small molecule is good enough or not, can be understood using the molecular docking method. And when I say that what should a drug do to be effective, this is a very interesting question, because a drug should actually have several characteristics. Okay, first, the drug should reach the site of action. Okay, so because I said that the protein that the protein is a receptor here, the small molecule must be able to diffuse somehow, reach the site of the action, that is a tissue or an organ if need be, and it should be able to bind to the receptor if it's a protein receptor or a DNA receptor, and it should be able to exert or display all the pharmacological properties such that the molecule is actually having the therapeutic effect. And most importantly, after the job of the drug is done, the drug should be able to leave the host within a reasonable time frame. That is, the elimination of the drug molecule is also important. And most importantly, the drug should not have any toxic effect. Okay, we are taking a drug because we want to be relieved of a disease, right? But then we do not want to have a new disease because of taking this particular drug molecule. We do not want to end up having a new disease altogether. Okay, therefore, not only should the drug molecule be eliminated, at the same time, it should also show non-toxicity to the human host. Okay, so when I say the drug molecule should leave the human host, maybe it should not leave the human host in about 2 to 3 hours, because then in a 24 hours time period, then the drug has to be taken 8 times. Okay, so if you say that the drug stays in the human body, say from 12 to 24 hours, then it means that you're taking the drug molecule for say twice a day or once a day. So it should be retained in the human body, so that the patient can take once or twice the drug, and it should leave the human host after a short period of time, such that we are not accumulating any new molecules or xenobiotics in our body, and this molecule especially should not have any toxic effect in the humans, right? So yeah, so when you say you want to study about the ADME basically stands for absorption, distribution, metabolism, excretion, toxicity. Okay, acronym is ADME. So this ADME can be studied at any stage of drug discovery. But again, we have to talk about the costs involved, and therefore, in keeping the cost in mind, most of the pharmaceutical companies or researchers go to the stage of ADME properties experimentally only after the molecule is validated using in vitro studies as well as in vivo studies. I think that is what the researchers do. But if you're doing computational work, as we are discussing today, my whole study is only about the computational study. So when you do computational study, as I said, you don't have to worry about what stage you should do ADME studies. In fact, if you do at the early stages of drug discovery, if you study the ADME properties of the molecules, it's actually good. And in fact, today there are many databases like, NCI data bank, Zinc database, so on and so forth. So all such databases have several millions of molecules. So if there's a student who's willing, then that student can actually spend a lot of time in studying the ADME properties of the molecules and come up with those molecules only that actually satisfy the ADME properties, so that you now have a subgroup of molecules from the Zinc database, and these molecules are actually able to are adhering to the ADME properties. Therefore, you can do the further docking studies and so on from that particular subset of database from the Zinc database, right? So as I said, you can go at any stage, but if you're doing computational study, maybe one of the projects for a summer student would be to download all the molecules from Zinc database and find out which molecules adhere to the ADME properties. And again, there are many websites which actually offer, like you can submit a set of molecules and then it will come up with certain parameters to say which molecules adhere to the ADME properties or not. So that's how, maybe some students can actually work on this project, right? So when I say absorption, what I mean by saying absorption is that how quickly, okay, before we go to the absorption, I'll say that there are over 100 methods of administering a drug. Maybe the most common ones are oral tablet form. Oral tablet form is actually good because the patient can take the drug himself or herself without any difficulty. They don't need the aid of somebody else. Intravenous form is another good strategy, but that requires the aid of somebody. Intramuscular form is another mode of taking the drug. And also, like, you have nasal drops. You can also take an ointment for local application. There are many ways, okay? And I would say that oral and intravenous are the most popular ones. And if you're taking intravenous, there is no problem of absorption because directly it is going into the circulatory system. Therefore, there is no issue of absorption here. The issue of absorption comes only for that molecule which is actually taken orally. So when you take a tablet orally, it first goes through your mouth, through the food pipe, it goes into your gastrointestinal tract. From there, it is taken up by the liver and other organs, and finally it enters the the circulatory system. And through the circulatory system, this molecule can go to the particular site. Okay? So absorption is an important parameter for the drug molecule, which is actually taken orally. Okay? And what about the distribution of the molecule? Okay, when I say the distribution of the molecule, what I'm trying to say is that distribution is the property by which the molecule is able to go to the site of action. So so that basically means how fast or how best it can be distributed from the circulatory system to the site where actually the molecule should perform, and what is the rate of distribution and what is the extent of distribution. So what is the rate of distribution is simple, how fast the molecule can be, can go to the site. What is the extent of distribution means, if you take 100 mg of the drug molecule, will all the 100 mg go there, or only 40 mg will go to the site and the rest 60 mg is distributed to the other organelles. Okay, so that is called as the extent of distribution. The more the drug goes to the site of action, to the particular location, the better it is. And when you talk about the metabolism, that is the drug is orally ingested, it has gone distributed to the site of action, and then the drug has to undergo a metabolism, that is biotransformation. That is, for example, the drug molecule will stay in the human host, say, for 6 to 8 hours. During this period, the drug will perform its action. At the same time, the drug has to be eliminated somehow. Before the drug is eliminated, most often the drugs undergo a biological transformation with the help of the enzymes in the host. And metabolism is in fact a very critical property for for all the drugs. And for example, say the molecule is lipophilic. Okay, it is if the molecule is a lipophilic, then the lipophilic molecule has to become somehow hydrophilic, and this hydrophilic molecule can only be that is a lipophilic molecule will undergo a biotransformation to become a hydrophilic molecule, and this hydrophilic molecule has to be eliminated. And there are many enzymes that actually play a role in this. And importantly, chemists, the first two steps, absorption, distribution, I would say that it's more of a biologist's work. But when it comes to the metabolism, after the metabolism is done in the human host, then there are biologists who actually study from which routes the molecule is being excreted. The molecule can come out from the sweat, it can come out from the tear glands, it can come out from urine, it can come out from fecal material. Sometimes the skin sweat also will also be able to excrete the drug molecule. Okay, so from such excretion, one will assess to study what are the various transformations that have happened to the molecule, and therefore one will try to understand what are the various metabolites of the original parent molecule. Okay, so for this, a chemist has to learn a lot of analytical tools. For example, HPLC is a very good method. GCMS, LCMS, NMR, mass spectrometry is also a very good analytical tool to tell what are the various metabolites that the parent drug molecule has actually undergone in order to before it actually is excreted from the body. And it's not that, and also there are some drugs like isoniazid, ethionamide, and some more such drugs. In fact, there's also one drug molecule for acne. I forget the name of it. But anyway, so these are called as pro-drugs. Okay, so this pro-drug is taken by the individual in the form of for therapy, and after the say ethionamide enters the circulatory system, then there are certain enzymes which will actually modify the molecule or the pro-drug into an actual drug, and therefore the drug actually becomes active. So it's not that it's not that whatever drug you take actually is a ready-made drug available for the function, but then what you take may not be active. But then there are certain molecules which will actually modify the drugs, and therefore a pro-drug becomes an actual drug, and the actual drug is able to do the function. So I would say that in the metabolism stage, there's a lot of role for chemists to play. And excretion, of course, is an important criteria, as I said, because you do not want the molecule to be residing in the human host for a very long time. It has to be cleared quickly. When you say cleared quickly, it should not be it's not like 3 to 4 hours, because then the patient should be taking the medicine every 3 to 4 hours. So it should stay in the human host for a reasonably long time. At the same time, not a very long time. So there has to be a balance between a drug that gets metabolized fast and not so fast. So yeah, so excretion tells you how quickly a drug can be eliminated. And toxicity, of course, is very important, because some of the drugs that we take are actually toxic to liver. Sometimes some could be toxic to our digestive system. Some could be toxic to the other organs in our body. Therefore, some will actually give you rashes. If you see the side effects of the medicine, they'll say that this medicine can actually cause rashes, skin rashes. Okay, so these all show that these are the toxicity. This is the toxicity profile of the molecule, and lower the toxicity profile of the drug, the better it is. So one would want to work with a molecule which is having less toxicity. But having said this, even a molecule that has got, say, FDA approval. In fact, Karunakar had worked on a molecule called as ponatinib, and this ponatinib was a FDA approved drug, and within 6 months after the FDA gave its approval, this molecule was retracted from the medical shops, because it showed a lot of toxic effects on which actually caused organ failure. So initially, while clinical trials are being done, it may not be known, these details may not become available. But then the moment it is known that this particular drug molecule is interfering the function of another normal protein, the physiological action of another protein, then you say that the molecule is showing a toxic effect, and therefore one has to withdraw it from the shelves. Okay. From what I have showed you in this slide, you will understand that we are talking about those methods which are completely experiment based, isn't it? These are all experimental methods, and you will wonder what is it that a computer can do in order to come up to understand what is the absorption of a molecule or a distribution of a molecule. How can one assess the ADME properties from the computational studies? Okay, so one of the first papers that came up was the work done by Christopher Lipinski in 1997. He worked for Pfizer, and it's also called as he came up with actually rules of 4, but because all the numbers are multiples of 5, this actually became Lipinski's rule of 5, or it's also called as RO5. It's also called as Pfizer's rule of 5. So he had actually examined the molecules that actually were available he had examined the FDA approved drugs, and he had come up with certain parameters and said that for a molecule to become for a chemical compound to become a drug molecule, it has to follow certain criteria, and these are the 4 criteria. It should say that a molecule should have not more than 5 hydrogen bonds, no more than I think while editing I removed donor here. It should not have more than 5 hydrogen bond donors. It should not have more than 10 hydrogen bond acceptors, and the molecular mass should be less than 500 Daltons, and the partition coefficient between the octanol and water, which is called as log P, should not exceed 5. Okay? So how did he come up with the magic numbers? So he came up with these magic numbers because these are not magic numbers, because there were lots. Okay, let me first complete. He has examined the molecules that were actually orally approved by, these were orally approved drugs by FDA. So he had taken up these drug molecules, and such analysis was made, and he has come up with these molecules. Okay, but you will see that there are many natural products which actually have a molecular mass more than 500 Daltons, but still they are able to function as good drug molecules. Okay. So though we say that this is Lipinski's rule of 5, you don't have to strictly stick to this. There can be certain violations to this, but more or less grossly the molecule should be able to follow this kind of a they should observe these kinds of parameters. And subsequently there were many modifications that were made to this. Okay, when I say hydrogen bond donors, what I mean is that hydrogen bond donor can be made by an electronegative atom, say such as oxygen and nitrogen. Okay, but then you can also say that there is a sulfur in my drug molecule. So what happens to sulfur? Does it are you going to take sulfur into consideration or not? Okay, so such considerations were made, and there were more variants to the Lipinski's rule of 5, like Weber's rules, and then you also have this Ghosh's rules, and there is something called as a fragment-based drug discovery approach. And in the fragment-based drug discovery approach, also such similar parameters were made. So okay, in the other parameters like Ghosh's parameters, you have something called as a molar refractivity of the molecule that was also considered. You can go back to see what is molar refractivity. That is not a part of the Lipinski's rule of 5. So this kind of a quest for looking at rules to find out what is the criteria that is required to make a molecule as a good drug candidate, okay, has come from theoretical studies. Okay, so though I have told you that all these are experimental methods, still you can apply theory to the known information, draw analogy, and from the analogy drawn you can apply it to the new molecules and find out if the molecule becomes a good drug molecule or not. Okay, so this I have taken from in fact, this is a paper by a Chinese group. Dong is the first author. They have come up with a online web server called as ADME Lab, where you give a molecule, you put the structure of a molecule, either the PDB, SMILES, whatever it is, it will come up with all these physico-chemical properties of the molecules. All right, but at the same time, one should be careful, because I mean, I always keep saying that the experiment is truth, and these are only indicators. Okay, so you should take such indicators with a pinch of salt. I wouldn't say pinch of salt because so far I said that the computer aided drug design is a good method. One has to, okay, though you get a positive result from the computational method, the experimental proof has to be there. Okay, I wouldn't say, sorry, I retract my word pinch of salt. You have to have the experimental proof for the computational studies. Unless you have the experimental proof, you cannot proceed further. Okay, so this is ADMET is once again a method that uses AI and ML to come up with the ADME properties of new molecules, which people have not studied earlier. And I would say that in the last 3 years, when I was preparing for this lecture, I've seen that there are at least about 5 to 6 good websites which will actually give you the ADME properties of the molecules, right? So yeah, so this is still an open challenge. You can still work on this, come up with newer methods. One possibility. The other possibility is that you can use these available methods to find out the ADME properties of the molecule of your choice, to find out if it has the desired ADME property, and therefore you can find out whether you can do the molecular docking studies, other biophysical studies followed by experimental studies or not. Okay. So before we go to the ADME properties, there are certain basic physicochemical properties that the molecule has to follow. The first one is called as log S. Here log S indicates solubility. So this log S directly tells you what is the extent of solubility of the drug molecule or a candidate molecule in water. Okay, it's as simple as that. And so what is the extent of solubility of the candidate molecule in water, and a log of it to the base 10 gives you the log S value. And the greater the solubility of the water, of the molecule in water, the better it is. But at the same time, you do not want to have a molecule that is completely hydrophilic, completely soluble in water. It should also have certain hydrophobicity, because these molecules, as you will see later, has to follow certain lipid bilayers or plasma membranes they have to cross, or the blood-brain barrier is what they have got to cross. Under those situations, you need to have a balance between the lipophilicity and hydrophilicity. But anyway, log S is one parameter, and again, depending upon the method that you choose, the log S values are in a range, and it's not one particular value. This log S can be given as, say, for example, moles per liter or milligram per liter. The units will remain the same, okay, because the concept is solubility in water, right? But then the range varies. So I'm not going to the ranges in my presentation at all, excepting in one or two cases. So log S tells you what is the extent of solubility of the molecule in water. And there is something else called as intrinsic solubility. That is log S to the base O, okay, to the subscript O. So that basically talks about the intrinsic solubility of the drug molecule in water. So there exists an equilibrium of the drug between the solution state and in the solid state. This usually happens in supersaturated solutions, isn't it? So, so the intrinsic solubility is the extent of solubility or the equilibrium that is established between the solution form and the solid state is called as the intrinsic solubility of the water. But I think most often you should be happy with So D here tells you gives you the extent of partitioning. Okay, what is the extent of partitioning of the drug molecule between octanol and a water molecule, and water, not water molecule, and solvent water, solvent octanol, solvent water. Why is octanol taken here? Because we always talk about a lipid bilayer. The lipid bilayer is supposed to have a fatty acid chain, and octanol with a C8 system is thought to be suitable enough in order to tell what is the extent of partitioning of the molecule between the polar and the non-polar phases, A, and also between to tell what is the extent of hydrophilicity or the lipophilicity of the molecule. So the partitioning of the molecule between octanol and water at pH 7.4 gives you the log D 7.0, and then you have log P 7. Log P, okay. So when you say log D, it is always at 7.4 pH is what perhaps you're considering. But then you can also alter this pH to say 6.7. Okay, why am I altering the pH to 6.7? Not 6. Let me say make it to 1.7. Okay, supposing say I take the drug and the drug has to go into my stomach first. Okay, so in the stomach the pH is low. The pH is very low when the when I'm on empty stomach. The pH say, like say 1.5 to 2, and say after I've eaten my meal, my stomach pH goes to 3. So you see that there is a difference in pH in my stomach versus another organelle, and the blood pH 7.0, okay, 7.4. So you can find out what is the extent of solubility at 7.4, at 1.5, at 3.0. All those studies can be done, and you can say that log D 7.4, log D 3.0. You can do all those studies, okay. And okay, so log P is a situation where you're only considering a molecule in the neutral form. See, when you take a molecule which has, say, charged side chains like carboxylic acid, primary amines, and so on, then their state will change. The protonation state will change depending upon the pH, isn't it? So in the case of log P, we are not considering such alteration in the pH. So log P and log D can be equal at the similar pH value. And under different pH values, the log P and log D are different. And I think it's log D that is most often considered because, as I said, of the changes in the pH from stomach to liver to the bloodstream. Okay, so these are the basic physical chemical properties, and then we go to the absorption of the molecule. That is, after somebody has taken the drug molecule, how quickly can the drug go via the bloodstream and reach the site of action. Okay, so one of the models that has been developed experimentally and also computationally is called as the CACO2 cell lines. So basically, CACO means cancer coli. So basically, colon cancer cell lines were taken from an individual and then they were immortalized. And this model is taken in order to find out what is the extent of the penetration of a drug molecule across the this colon cancer cell lines. And there are methods computationally which also will tell you what is the extent of permeability of the molecule across a cell membrane. And again, as I said, I'll not give you the figures, but then generally it is considered that a molecule should be able to penetrate well enough such that it's able to cross the barrier. I mean, from one cell to other cell, if the molecule has to diffuse and enter, there are two possibilities. Okay, one of the possibilities is that the molecule will have a passive diffusion. When I say passive diffusion, what I mean is that the lipid bilayer or a plasma membrane is impermeable. It can only allow certain amount of molecules to come and certain amount of molecules to leave, isn't it? So there is one possibility is that. Are you all able to hear me?
Yes, ma'am.
Thank you. So one possibility is that there is a passive diffusion. The molecule is able to simply pass through the plasma membrane. That is that's an easy situation. And the difficult situation would be the plasma membrane on the human cells will have certain transporters, and these transporters are usually like, say, potassium channels, calcium channels. They can also be glucose transporters. Likewise, such transporters are also allowing a drug molecule to diffuse from one location to another location, from outside the cell to inside the cell. Okay, so such experiments need to be done to tell what is the extent of permeability of the drug molecule to the site of action. And there is something called as a PG. So it also tells you the speed. Okay, the greater the speed, the better it is. That is, the molecule is able to reach the site of action. I mean, if you take the drug, it should not show effects after 3 to 4 hours, isn't it? The sooner the molecule will show its therapeutic effect, the better it is. So yeah. So CACO2 is, as I said, it's one kind of immortalized cell lines from colon cancer cells, and this basically tells you what is the extent of the absorption of a drug by a cell. And when I say PGP is basically a P glycoprotein or a phosphoglycoprotein. In fact, this is also a transmembrane protein. P glycoprotein is also a plasma. It's also a lipid bilayer protein. It spans from outside to inside, so it's called as a intrinsic protein, intrinsic transmembrane protein. This is present in various type of cells such as liver, kidney, gastrointestinal tract. Several, in fact, the blood-brain barrier also has these kinds of PGP phosphoglycoprotein, and the drug that you take, and and what is the physiological role of this molecule is, this molecule is able to function as an efflux pump in order to allow certain molecules to go from inside to outside, outside to inside. Okay, so but one would want to understand whether the drug that they have designed is acting as a PGP inhibitor or if it is acting as a PGP substrate. Okay, if it is working as a PGP substrate, perhaps you're in a good shape because because of the because of the PGP protein, it's able to penetrate inside the cell. But if this acts as a PGP inhibitor, okay, if the new drug candidate that you have acts as an inhibitor, then you can think that the physiological function of the protein could be lost. Therefore, one would want to establish whether it is an inhibitor or a substrate for the P glycoprotein. And the other thing that one would want to study is HIA. HIA basically stands for human intestinal absorption. Again, human intestinal absorption means that what is the extent of the absorption of the molecule by the gut. Only if the stomach is able to absorb the molecule, only then it can go through the various parts of a body through circulatory system, isn't it? Therefore, it is very important to understand what is the extent of human intestinal absorption of the drug molecule, and also there is an extent of numbers given to this. Okay, if the absorption is 20%, 30%, not 20, 30, is it below 20%? Is it between 30 to 80%, or is it above 80%? Okay, so these three parameters are given, and if the molecule lies, say, between 30 to 80%, then you say that the molecule is a good drug molecule, or it's a good drug candidate to be absorbed by the gastrointestinal tract. And the third, I think I have, I'm so sorry, I have them all here. Right. Yeah. So, okay, that's all right. So the third aspect, which is distribution, talks about the PPB, that is plasma protein binding. You know that in our blood we have, in addition to the red blood cells, we also have this plasma proteins, and these plasma proteins are very many in I wouldn't say very many in number. The most important plasma protein, I think, is the serum albumin is one protein, that is a plasma protein. In addition, you have certain glycoproteins, you have certain lipoproteins, and then there are certain proteins called as alpha, beta, and gamma globulins. So you would want to understand what is the extent of binding of the drug molecules to each of these plasma proteins. And it's like this, okay? You would want the molecule to go through the bloodstream, yes, but then if it is able to bind to the plasma protein very effectively or with high affinity, then the possibility of it being delivered to the site of action is less, isn't it? Because the drug candidate is having greater affinity. Let us say talk about serum albumin. Okay, most people do these studies. If the small molecule has a high affinity to the human serum albumin, then it may not detach from the serum albumin and go to the site of action where it is required to function. Okay, so it should bind, but then at the same time it should not bind to the serum protein with very high affinity. If it is able to bind to the serum protein with very high affinity, then it basically means that it's not a very good drug candidate. And then you also have something called as a VD, or volume of distribution. So volume of distribution once again talks about the amount of drug that is present in the body in milligram quantities divided by the plasma concentration of drug. That is how much of the protein is actually bound to the plasma proteins. Okay, I just said that you have this lipoprotein, glycoprotein, you have the globulins, and you also have mainly this serum albumin. Okay, So what is the amount of drug in the body that is unbound versus what is the amount of protein that is bound to the plasma is also calculated, and it's called as the volume of distribution. And again, if you have more amount of drug that is floating and less amount of protein, if you have more amount of drug that is freely flowing along with the blood and less of it is bound to the plasma protein, then you say that that particular drug becomes a better drug candidate. And then you have and then this blood-brain barrier is once again again, when you talk about blood-brain barrier, you're not talking about, I mean, you already know that we're talking about somewhere about the brain, we're talking about neurons. See, it is very, see, our human body is very intricate, but then it is also intricate, delicate, but then it's also very robust in many sense, okay? Because not every molecule will reach every site, okay? For example, say there is a viral infection, and the viral infection should not reach the brain for many reasons, okay? Therefore, there are many molecules which will actually not cross the blood-brain barrier, for example, the virus molecules. But then there are certain molecules, like for example, small organic chemical molecules, which are required to cross the blood-brain barrier, okay? For some reason, okay? Or you have some molecules that should not cross the blood-brain barrier because they will cause toxic effects to the brain, for example, okay? So you can also understand what is the permeability of the molecule across the blood-brain. And again, we were talking about gastrointestinal tract cells, okay? When we talked about, here we talked about, Caco2 cells are from the colon cancer cells. And here we said that PGP is present in many cells, like say liver and so on. And human intestine, this is only present in gastrointestinal, okay? So one is trying to study what is the effect of, or what is the ability of the molecule to permeate through various lipid bilayers. And here also, depending upon the role of the protein, it can or it cannot cross the blood-brain barrier. And when it comes to metabolism, again, I said that metabolism is something that is quite important for somebody to understand what is the extent of changes that occur in the molecule not just the extent, but then what are the kinds of changes that will occur in the molecule before the molecule actually becomes, stop performing its action, or before the molecule actually is ready to leave the human host, okay? So the changes that occur in the blood, the changes that occur in the the changes that occur to the drug molecule or the likely drug candidate in the human host are done by several enzymes, and the most important family is called as the cytochrome P450 family of enzymes. There are about 250 cytochrome P450 family of enzymes in humans. And if you see my list here, you say that it is looking at cytochrome P1A2 inhibitor, cytochrome P3A4 inhibitor, so on and so forth. You only have about how many? 10 enzymes here. So the other 250 enzymes are also there. But so far it has been seen that these molecules are able to, these 10 enzymes are perhaps able to take part in about 80% of the metabolism of the drug molecules. So therefore these molecules are are effectively being studied. See, cytochrome P450 is a wonder enzyme. It has a porphyrin ring, and the importance of this enzyme is that it's not only not only waiting for drug molecules, okay? Even if you take say some food that we take, okay, the food that we take is also acted upon by the enzymes in the cytochrome P450 superfamily of enzymes. And if suppose we say we take some herbal concoction, okay, or some xenobiotic, all such molecules are also metabolized by the cytochrome P450 family of enzymes. And therefore you would want to see, because these enzymes are able to perform a regular physiological function, you do not want to inhibit any of these family of enzymes. In fact, if you read any metabolic metabolism paper for a new drug molecule, they will at least mention 3 to 4 different cytochrome P450s which are able to metabolize the drug molecule. For example, one cytochrome P450 is able to break a CC bond. There is another cytochrome P450 which is able to add a OH group. There is another one which is able to convert a carboxylic acid to a aldehyde group, so on and so forth. Okay. So there is not one single enzyme that will act on a drug molecule, but then there could be 3 or 4 cytochrome P450s that will act on the supposedly drug molecule. But then you would want to understand which drug molecule, which enzyme has metabolized which drug, and what are the metabolic, it's called as metabolic profile. That is, you collect all the metabolites from, say, the urine sample. If somebody is, say, given a drug molecule, then the urine sample of them is collected after 8 hours. I mean, at regular intervals, the samples are collected. And from that urine sample, one can go back and find out what are the various metabolites which were not present before this candidate has taken the drug molecule. And from these metabolites, a metabolic profile is made. From this, one will be able to find out what are the alterations that have taken in the molecule, and therefore one will be able to tell which are the cytochrome P450s that have actually acted upon the metabolite. And of course, having established that, there are many people have actually cloned all these enzymes, and therefore one can do a enzyme assay very quickly. Yeah. And then the next one is the clearance rate. Okay. So what is the clearance rate here? You would want to understand how quickly the drug is eliminated from the bloodstream, how quickly the plasma is no more bound to the drug molecule, is called as the clearance rate of the drug molecule. But as I said, it's not, it should be there for sufficiently long time, but then it should not be there for too long a time. And then, okay, as I said, some of the drug molecules also will it will not be a drug, but then sometimes a metabolite will also create a can also create a injury to the, say, for example, liver. It's called as drug-induced liver injury DILI. So you'd want to study, because you don't want to take such molecules, as drug molecules, isn't it? You don't want to be getting rid of, say, fever, but ending up having a liver problem, isn't it? So one will want to study what is the extent of liver toxicity caused by these molecules. And then one will also study something called as a Food and Drug Administration recommended maximum daily dose, FDA MDD. See, we can take, say, for example, few milligrams of drugs, all right. For example, say if you take paracetamol, we take something like, say, 600 mg of paracetamol, isn't it? If you have very high fever, that is still all right. But you should not be end up taking 2 to 3 grams of a tablet, isn't it? That is 2000 to 3000 milligrams of medicine nobody would want to consume. Okay. So from this, if the studies say that the drug is getting released very fast, because the drug is getting released very fast, we should take the drug more frequently or more periodically. And if this ends up to, say, large numbers of milligrams of the drug molecule, then it is not recommended. Therefore, one will also study what is the extent of toxicity, how quickly the drug is released. Does it cause any injury to the liver? Does it cause any injury to other organelles also, I'm sure. And liver is more importantly, because all your that is a reservoir of enzymes where the all the metabolism takes place. So you say that you want to study the injury to liver. Likewise, you don't want to be taking large amounts of drug molecule, right? And then, when we talk about toxicity, earlier I said that there are certain drug molecules which actually were taken initially by certain individuals, but these individuals actually ended up having different symptoms, and therefore the drug had to be removed from the market. Okay, one of them is called as the actually called as the HERG. It's called as some endothelium. If I remember, I'll come back to this again. But basically, it is a cardiac potassium channel, and if the drug molecule is able to bind to these particular potassium channels, then it actually causes heart attacks to the patient who has taken it. Likewise, if it also binds to this hydroxytryptamine receptor, also it causes heart problems, and therefore you do not want to have a molecule that is causing toxicity to various other organelles, liver here and heart problems later. And then skin sensitivity, you don't want to be having rashes or itches after you have taken the drug. And also they study something called as the acute toxicity. Okay, so what is acute toxicity? Again, these are done in the phase 1 of clinical trials. So that basically shows what is the extent of adverse effects of a substance that results from a single exposure of a drug. Okay, so you either take, say, about 10 grams of the drug and see what are the various adverse effects that the individual will see, or within a short span of time, like say within 12 to 24 hours, several doses of the same drug is given. It is as good as a single exposure, but multiple exposures within a short period of time is given to see what is the acute toxicity of the person. See, when we take a drug, we take small doses over a long period of time. Okay, maybe 3 times a day paracetamol for 4 days. Okay, that's one treatment regime. But then if you have to say take about 3000 milligrams for 3 times a day for 10 days, okay, then definitely that is not recommended, and it comes under the this FDA MDD disapproved treatments. Okay, so one would want to study what is the acute toxicity caused by the drug molecule under various conditions. So again, I keep saying that these are all the experimental methods, but having said that, from the known molecules and the toxicity profile, from the ADME properties profiles of all these molecules that are documented, one is able to come up with very many parameters. Okay, for example, there is a case study for a drug molecule which talks about this HERG cardiac potassium channel. From drugs, about 37 different parameters were taken, not one drug, from a set of drug molecules that were tested. 37 parameters were delineated. These 37 parameters were fed into were fit into machine learning, and from this and then you see which molecule is good, which molecule is bad, and based on these 37 property profiles of many molecules, one is able to tell which features are good, which features are not good in the molecules. Also, in the case of toxicity, it is already known that there are certain functional groups, there are certain there are certain functional groups or linking regions. If these linking regions are present during the metabolism of the protein, these these new functional groups are created, which actually cause toxicity to the individual. Therefore, there are also a list of linkers or functional groups which should not be present, because these are bound to cause toxicity to the patient, and therefore such functional groups are actually not recommended. So though it is largely an experimental based method, because there's a lot of information already known today, so you keep the information that is known, come up with certain desired parameters. These parameters are good, these parameters are not good, and therefore you're coming up with some sort of a QSR model. And this QSR model, you're able to map it onto certain new unknown molecules. That is where one can actually apply AI and ML to the new molecules that one can come up with, and decide whether these molecules are good or not. And therefore you can decide which molecules to prioritize in terms of your experimental methods. So this brings me to the closure of the ADME properties of the molecules. And now I will discuss with you about the molecular docking. Okay. In the case of molecular docking, the molecular docking, I think, is quite a straightforward subject, in the sense that you are not, okay. I wouldn't also not call it so straightforward, because earlier people would only try to dock those molecules which were synthesized in their laboratory only. But that is not the story anymore. But then people are trying to dock libraries of molecules which say have about millions of entries. Okay, so that is a challenging task, but still this is possible today, because the ease of access to small molecule libraries is there. There are many companies which actually give. Select is one such company which gives access to the millions of compounds library. But then the good thing is that if we find any of those molecules to be good from computational studies, then we can go back to them and buy those chemicals from them as a vendor. Therefore, they're getting business, and then we can do our experimental methods with them. We can do our experiments with them. Okay, so we have ease of access to such large libraries of molecules, and today we have protein databases. And as I said in the earlier, we have this AlphaFold, which actually has deciphered the structures of several millions of molecules. So we don't even have to model the structures of proteins anymore. And importantly, we have reliable software. Okay, we have reliable software to tell whether the docking is good enough or not. And when I say we have reliable software for docking, as well as assessing of docking to tell whether the docking is good enough or not. And then we also have, in order to host this good software and also in order to access these millions of compounds libraries, we have very powerful computers. So, these are the reasons why we have, why we are able to come up with very good, very quickly we're able to do the molecular docking. Okay. So what happens in the case of a molecular docking? So in the case of molecular docking, one is trying to predict, okay, what is the structure of the intermolecular complex form. All right. So to start with, you have the structure of a protein, let us say a receptor, which has a cavity into which a small molecule is likely to bind. And you also have the structure of the small molecule. So now you want to know what is the context in which the small molecule will bind to the binding site in the receptor. Okay, it's as simple as that. So here we are trying to say what is the preferred orientation of the small molecule in the binding cavity of the protein. First question. And in order to allow the small molecule to bind to the active site of the enzyme, there is also something called as an induced fit model. The protein active site also undergoes a slight conformational change. And there are two methods for docking, more methods actually. One is called as a rigid docking, the other is called as a flexible docking. So if you do flexible docking, you have alterations in the small molecule conformation. You also have alterations in the protein active site, right? And when you say that a stable complex is formed between the protein and the ligand, then the stable complex is stabilized by certain non-bonding interactions, and therefore these non-bonding interactions also, the greater the non-bonding interactions, the better is the docking, right? And right. So this is a very primitive way by which we can show how molecular docking is done. For example, this is the target selection. That is, you know what is the protein with which you would like to work with for a disease state. For a disease, there could be more than one protein, but then you have one protein which is say overexpressed under a disease condition, and maybe your laboratory has already established the experimental methods for that particular enzyme. Therefore, you will take that as a target protein. And then when you say preparation of the target, I'm sure you have learned all these things before, or you learn in the next few days. Here, what happens is you would want to, in the protein data bank, in the protein structure, you don't have hydrogens. Hydrogens are added. Water molecules are removed. Then certain heteroatoms are removed. And if a ligand is already bound, that ligand is removed. Sometimes you will have let's say polyethylene glycol, glycerol in the crystal structure. You remove all those things, you prepare the protein, and often you would want to know under what conditions the protein is active, and therefore you will add hydrogen, say at pH 7.4, at pH 9, pH 3. You do all those, you do the protein preparation, and then this is what ligand selection, right? So when you say ligand selection, what you can do here is maybe you have 20 molecules from your lab which you have synthesized, or you have a library of compounds. It's up to you. So you basically have several. Let us say you have a compound library with say 10,000 molecules. You take those molecules again. These are usually in a 2D format. You convert 2D to 3D format. You add hydrogens, and there is no one unique conformation to the protein, to the small molecule, right? Because I'll show that in one of the subsequent slides. When you say that the protein will have one unique conformation, okay, protein also will not have one unique conformation. Protein also has dynamics to the protein structure. But then let us say that the active site is stable, okay? The ligand here will have several rotatable bonds. So because of rotation about several of these rotatable bonds, you don't have one unique conformation to the ligand. Therefore, you should be able to generate many conformations to each of the ligand. So a molecule, say of 20 molecule library, will end up having 400 different conformations. And then, okay, all that comes under ligand preparation. And now you dock the small molecule into the active site of the protein. That is called as molecular docking. That is, you're trying to see how well a small molecule fits into the active site of the enzyme. And then you will evaluate the docking results in terms of scoring. Okay. Scoring is one way of looking at it, and most often the correct way. Often you will also do a manual visualization to see, supposing say from your earlier studies, you know that certain residues in the active site are important, and some experimental paper says that these amino acids are important in the active site of the enzyme. Then from your docking, you want to see whether those regions of the protein are able to make any interactions with the enzyme active site or not, for example. Okay. So you will find out, you will take the quantitative score. At the same time, you will also visualize the protein ligand complex, and then you will evaluate the result to see whether you've got the right solution or not. Often you may not get the right solution. There's some theory called as junk in and junk out. That is, if you have not been careful enough in any of these stages, you may not have a good docking pose at all. That's one possibility. And the other possibility is that you will see that there are at least about 10 docking methods that are freely available to a user in the public domain. So not all methods will work for all protein systems. Okay. So if one method fails, you should go to the other method. Or importantly, you should try at least 3 or 4 docking methods to see which methods are able to give you reliable results. Or you can also look at something called as a clustering of the docking conformations also you can do. That is, supposing say you see that out of you say one molecule is able to dock in the same pose at the same location 8 out of 10 times. Okay, then you say that it is a then the clustering of that molecule is very high, because more times the same pose is repeatedly coming, and therefore you give more emphasis to that. So there is no one way by which you can say which method works and which method does not work. And I think it comes more with practice. It comes more with practice. Right. Yeah. So okay. So when you have prepared a target and when you have done the docking, where is it that you will do the docking? Okay. This one should have actually come first. But anyway, if there is previous information, such as if the protein structure has been solved in complex with a ligand, or if a homologous protein has a ligand or a cofactor bound at the active site, or if somebody tells you what are the active site residues, if somebody says that upon mutation of one particular amino acid, the activity of the enzyme is lost, then you will believe that that is the active site of the enzyme, and your docking should be done at that particular place only. So this becomes a region of your interest, and then you will do the docking there. So basically, here you are doing the simplification of the search problem. It's called as targeted docking. I did not mention here. But sometimes you will not know what is the active site of the enzyme. That is, only the structure of the apoprotein is solved, or perhaps you don't have a template structure for the protein, or the structure of the protein has not been solved. You have just done the modeling of the protein. And when you do not know what is the active site of the enzyme, then there are very many methods to tell you what are the binding cavities in the protein. Okay. It's again an area by itself. One can find out what are the various cavities of the protein surface, and therefore one is you can find out the cavities, or what you can do is you just don't be in the absence of a known cavity, you can do something called as a blind docking. That is, you consider the entire protein as a cube or as a sphere, and then see what are the various crevices or pockets on the protein surface where these small molecules can bind. Okay. In the absence of known information, it's called as a blind docking. When the information is available, it's called as targeted docking. Right? Yeah. So I've already told you how you do the ligand preparation. You do the ligand preparation from various download molecules, zinc, NCI database, PubChem, so on and so forth. Or you can also sketch the molecules using ChemSketch. And then I'm not keeping any particular methodology here, because as I said, all methods are good, all methods are bad. It depends on how persuasive you are to come up with the right answer. All right. And most often, how do you know that a docking is good? You take a known protein ligand complex from a homologous system, and you do the docking once again. If it is able to give you the crystal structure confirmation, then you can believe that, yes, this particular docking method could be good enough. And yeah, so the ligand is docked onto the receptor and interactions are checked. The scoring functions generate a score depending upon the best fit ligand is selected. So what, I don't think I have anything here. I don't have anything about scoring functions, but anyway. So scoring functions basically depend upon the extent of, the more the attractive forces, see when you say about the non-bonding interactions, you can have attractive forces as well as repulsive forces, isn't it? What are the attractive forces? I say that I have ionic interactions, a positive and negative coming close to each other within a certain distance, okay? Again, within the same distance, if one positive and another positive are coming, then I would say that it is a repulsion. Likewise, if I say that a negative and negative are coming close together, charged two atoms, one from protein, one from ligand, then I say that there's a repulsion. Likewise, you have Van der Waals forces of attraction, repulsion, so on and so forth, okay? So here we can give the scoring function can be weighted by the summation of the attractive forces, and then you can also add a penalty to the repulsive forces, and you can also add a penalty to any steric to any not steric, what do you call strain, okay? If the small molecule, see when you take a small molecule, the small molecule can have n number of conformations, and a conformation that is having the if the molecule is say docked in the active site of the protein with a certain amount of strain, okay, then the strain also can be penalized, or one can actually add a penalty in the scoring function, okay? So this is how the scoring functions work anyway. So the success of a molecular modeling depends upon many factors, and the important factor I would say is the conformer generation, okay? I would say that I would want to have the pose or the binding pose in which it is able to bind to the active site of the enzyme, okay? But supposing say during my molecular docking, if the molecule does not go through that conformation at all, then the chance of me finding the correct docking pose will not arise, isn't it? Because the molecule is supposed to have a certain amount of bend, it's supposed to have a certain amount of torsion angles about various atoms, so on and so forth are required, okay? But during the ligand preparation, if the molecule has not witnessed such a conformation at all, then the success of molecular docking will not happen at all, isn't it? So the conformation generation is a very important aspect in molecular docking. And then there are many methods. This again depends upon the method that you're using in terms of your molecular docking package. You have a systematic docking, that is you take a small molecule, and then you have many rotatable bonds. You know that rotation about double bond, triple bond is not possible. Rotation is possible only about a single bond, isn't it? So therefore what one can do is you can slowly increment the rotation about each of these atoms in this small molecule, and you can generate a variety of conformations. That is one possibility. That is a systematic search method. The other is called as a stochastic search method, where you are randomly modifying the structural properties of the ligands. At the same time, you would want to avoid. At the same time, you would want to avoid a conformation. At the same time, you would want to avoid a conformation which is, one second. At the same time, you would want to avoid a conformation which is actually trapped in a local energy, okay? What do you mean by local energy? Supposing say the molecule is trapped somewhere here, and then does not have enough energy either to look for a conformation on either side of the energy barrier, then the other conformations will not be achieved at all, isn't it? So you would want to have a molecule that is likely to have a global minimum energy, and that is not supposed to be trapped in a local energy minima, okay? So the stochastic search methods work that way, and both methods are reasonably good. And the stochastic search methods are faster because they're not doing a systematic search, of every rotatable bond. So that way this method is, stochastic search method is faster compared to a systematic search method. And definitely there's a possibility of finding a good, global minimum energy conformation of the small molecule also. Yeah. So yeah, I told you very briefly how scoring functions work. Scoring functions basically try to give you a good assessment of all the good non-binding interactions as well as the bad non-bonding interactions. Their summation gives you the scoring function, gives you a score and tells you how good a docking poses. And again, scoring functions can be empirical, ab initio, force field based, knowledge based, many types. And again, I mean, though I say that an accurate docking method will identify correct pose with a higher score. Okay, there is nothing like a accurate docking method. It just doesn't happen. You have got to work with several methods and find out which method works for the system that you're working. Supposing, say, if you're using gold as a methodology for docking for one system, then just because you have learned it, you cannot just apply it. You have got to still work with autodock, glide, whatever fire, what is that? The other methods also, autodock also, to find out whether this method works for you or not, and evaluating the docking results. So as a rule of thumb, yes, you can take the scoring function as a method for to find out whether the docking is good enough or not, but that is not a gold standard. It, as I said, it comes with a certain amount of experience. So you should read the theory papers. You should read the theory papers in terms of methodologies. You should learn about the active site of the enzyme by looking at what the crystallographer has to say, or what the authors of the homologous papers say. And keeping all these parameters in mind, only you should be able to evaluate your docking result and decide whether your docking is good enough or not. And as I said, if the docking is not good, because see, when you're doing experiment, you have the fear of wasting reagents, chemicals, and therefore you will also feel that your supervisor may not be happy because you have wasted a lot of resources. In the case of computing, unless you're doing molecular dynamics, you're not wasting any resources. Otherwise, you're doing most of the work on personal computers only or on desktop only. So you're not wasting any resources that way. All that you gain is experience by repeating a method twice, thrice, 4 times, 10 times, till you are able to get a result that is good enough. And how you know whether your result is good enough or not, depends again on other aspects. Okay, so with this, I stop, and I acknowledge the School of Chemistry, University of Hyderabad, for for the employment, for all the benefits that I get, and all the wonderful students with whom I get to work, and with whom I have learned many things. And then I also thank the organizers of this National Faculty Development Program, both Reddy College and Q Statics Purushottam and Dr. Purushottam and Dr. Karnataka. Thank you so much. And I'll be happy to take any questions if you have.
Thank you, madam, for your wonderful presentation. Dear participants, if you have any queries, you can unmute yourself and ask ma'am.
I'll stop sharing so that I might be able to see them. Is that
yes,
okay?
ma'am. Yes,
Ma'am,
ma'am.
I have a doubt, ma'am.
Yeah, sure.
Yeah. How reliable is this blind docking, ma'am, during the drug designing?
It's actually good. I mean, it's like this. Okay, when you do a targeted docking, where the active site is. Where the small molecule is likely to bind. Okay, but at the same time, this need not be the site for this particular molecule. You have some template protein, you have some binding site, and you believe that this small molecule has to bind here, but it need not be necessary. It's like telling that there is an elephant with big trunk, 4 legs, and trunk is the one that the elephant might use to eat. So that is as far as you can go in terms of a targeted docking. When you do a blind docking, you just don't know anything about the elephant. You just know it's huge and it's huge big. But having said that, I think there are two projects in which we have done blind docking and we have been quite successful. One is a G quadruplex DNA, and the other is a same bovine serum albumin. And for both, we were quite happy with our results. Yeah, you use blind talking only if you have no information about the protein target. You don't know where the small molecule is likely to come.
Thank you, ma'am.
Yeah, Ambuja, you can ask your query. Hello.
Prolonged use of salicylates as a science is that the issue?
Any questions?
Ma'am, there is a question from Ambuja.
Right.
Prolonged use of salicylates cause ulcers. Why?
I really don't have answer to this question. Maybe it is, we have seen so many properties, right?
Yeah, ma'am.
I'm not the right person to answer this question. I'm sorry.
Still, anybody is having doubts, you can ask. Yes, still anybody is having queries? Yeah, if not, we'll end the session. Madam, you can stop sharing the screen, ma'am.
Yeah, I just stopped and I've come back to I am seeing more people now. Yeah, I've stopped.
Yeah.
Thank you so much once again for the opportunity given.
Okay, thank you, ma'am. From Department of Chemistry and Forensic Science, we would like to present a letter of appreciation to our guest of honor and keynote speaker, Professor Lalita Guru Prasad, Madam, School of University of Hyderabad.
Thank you, ma'am. I accept this.
Now, I request G. Vijayalakshmi, Assistant Professor in Department of Chemistry, to propose vote of thanks to our keynote speaker. Over to you, ma'am.
Thank you, Shravanti. Good evening, one and all. On behalf of Department of Chemistry and Forensic Science, I, G. Vijayalakshmi, Assistant Professor, feel proud and privileged to propose vote of thanks to guest of honor and keynote speaker, Professor Lalita Guru Prasad, Madam, School of Chemistry, University of Hyderabad. Madam discussed about the ADMET data at early stages of drug discovery by explaining very clearly absorption, distribution, metabolism, excretion, and toxicity, and also the importance of analytical tools to the metabolism process, and Lipinski rules for an orally active drugs, and also different parameters of ADMET lab stages. Madam also explained how molecular docking useful to predict the structure of the intermolecular complexes formed between the two molecules, and also rigid docking and flexible docking and conformer generations, and the important aspect of the molecular docking, that is the conformer generation. And with the deep sense of gratitude, I thank you, ma'am, for delivering an interesting and invaluable lecture. We all are benefited by your lecture, madam. Thank you very much.
Thank you, sir. Thank you, ma'am.
Thank you, ma'am.
May I now log out? I think the session is over, right?
Yeah, ma'am.
Thank you so much.
Thank you, ma'am.
Thank you.
Requesting all the participants to fill the feedback form. It is posted in the chat box. I request the participants to fill the feedback form, and the link is posted in the chat box. Tomorrow we'll continue with the second day of the session. It starts at 2 p.m.